Good afternoon. I am Gino Nicolais, Chairman of the Scientific Committee of the Città della Scienza IDIS Foundation. And I thank you all the speakers that today will be with us, and especially our scientists that they agreed to just spend some time in this transmission. I must say that this meeting has been organized by the Italian Embassy in Stockholm, in Sweden, and Mattia Mazza, that is the embassy representative, will just say a few words about it. Please, Mattia.
Dear President Villari, dear Chairman Nicolais, dear Professor Masucci and Professor Mantovani, dear Ms. Bencivelli, dear all. It is a great pleasure to gather virtually here today to participate in this important event, together with such prestigious speakers, talking about viruses and their evolution, focusing on the cooperation between Italy and Sweden in this field. For this, allow me to extend my deep gratitude to Città della Scienza di Napoli for their precious cooperation and extraordinary national and international value in promoting scientific knowledge and expressing some of the best examples of the extraordinary quality of Italian research. As you may know, today's event takes place within the framework of the Italian Research Day in the World, an initiative that aims to cast light on the Italian excellence in this field of science and technology, as well as on the work of Italian researchers alongside the results of their research throughout the world. Italian embassies, consulates, and cultural institutes around the world are all committed to supporting this initiative and celebrating this event with the aim of showcasing the very best that the Italian research and innovation system has to offer. To this end, a solid network of amongst universities, research centers, and industries is of paramount importance as it paves the way to the development of the technological know-how. As the President of the Italian Republic stated recently, the Constituent Fathers placed the promotion of scientific research among the objectives of the then newly born Italian Republic, an understanding founded on the unity of humanistic, scientific, and technological knowledge, which is and was particularly far-sighted back then, and it is all the more today, given the current situation. In this context, I believe it is important to underline the Italian concrete commitment to support cooperation amongst universities, exchange programs for professors, and scholarships, which enable students and researchers interconnectivity and, when possible, mobility. Italy and Sweden are already going along this path and are committed to do more and better. Italy strongly believes that international university cooperation is a powerful tool to promote academic freedom, and it also represents an extraordinary lever for technological and economic development. As far as Italy is concerned, figures testify to the intensity and geographical extent of these collaborations. We have over 17,000 cooperation agreements currently in force between national universities and research centers and foreign counterparts. In this framework, inter-academic cooperation between Italy and Sweden is remarkable because we have 186 bilateral and multilateral agreements. Our embassy, in this respect, seeks to be more and more a facilitator of active connections amongst universities and research centers for a broader variety of research projects. Something that, together with Swedish authorities, we're all advocating for, as proven by the many projects supported in the framework of the ongoing executive program for scientific and technological cooperation between Italy and Sweden. And further, through a series of initiatives that the embassy organizes within the scope of integrated promotion. For instance, the very successful Italian-Swedish Health Summit on diabetes and cardiovascular diseases organized in 2019 here in Stockholm. Sweden and Italy closely cooperate and deepen relations through the years owing to their shared vision of their strategic interest in the development of knowledge. It is within this framework that we are developing our bilateral relations with a strong sense of friendship and partnership. Academic exchanges and collaboration generate demonstrate truly shared benefit both for researchers and technological development, and for the purpose of a greater economic integration. At the same time, they are functional to the national soft power, to which both our countries have a lot to share and learn from each other's perspectives. Having said all that, let me just finish by extending my acknowledgement to all the Italian and Swedish scholars and researchers connected with us today, and express them my sincere wishes for their best success. And now it's time for you to enjoy the video made by Barbara Galavotti, Italian researcher and scientific communicator, on the occasion of the Italian Research Day in the world. Please enjoy the video.
Oltre 30.000. Questo è il numero dei ricercatori italiani che lavorano fuori dal nostro paese secondo le stime del Ministero degli Esteri. Negli Stati Uniti siete circa 15.000, 6.100 nel Regno Unito, 4.000 in Francia, ma siete ovunque. Un piccolo manipolo in Corea e poi in Argentina, Sudafrica, India. Non c'è un continente in cui la scienza non progredisca anche grazie a ricercatori italiani. E questo è naturalmente motivo d'orgoglio, ma non è una novità, perché la ricerca per sua natura è internazionale, non conosce confini. Da sempre, oltre alle idee, hanno viaggiato le persone, a cominciare dal genio per eccellenza, Leonardo, che nel 1517 si trasferisce in Francia, dove continuerà i suoi studi di ingegneria, anatomia e sulla natura. Ma se la scienza antica ha in comune con quella moderna curiosità e passione, ben altri sono i mezzi di cui disponiamo oggi, mezzi tecnici naturalmente, ma soprattutto il più potente di tutti, l'unione delle capacità mentali. Perché se la scienza antica era caratterizzata da geni solitari, quella moderna trae la sua forza dalle grandi collaborazioni, come quelle che sostengono la fisica delle particelle al CERN o la grande ricerca spaziale coordinata dalle agenzie internazionali, fino alle piccole collaborazioni fra ricercatori che si stabiliscono quasi spontaneamente per indagare problemi specifici, come alcuni di quelli che hanno a che fare con l'epidemia che stiamo vivendo. I ricercatori italiani all'estero sono allo stesso tempo parte della comunità scientifica del nostro paese, di quella del paese che li ospita e della comunità internazionale largamente intesa. Ora, per il bene di tutti noi che dalla ricerca ci aspettiamo conoscenza e soluzioni, è importante che queste tre identità convivano senza affievolirsi. È fondamentale che la nostra nazionalità continui a essere una risorsa per chi fa ricerca all'estero, ed è cruciale che la grande qualità dei ricercatori italiani sia percepita all'estero come la naturale conseguenza di un'eccellenza scientifica che non venga fraintesa per una somma di talenti individuali, cosa che sarebbe inverosimile vista la vostra ubiqua e numerosa presenza. Per tutto questo c'è l'impegno degli addetti scientifici e della rete diplomatico-consolare, c'è il supporto di Innovitalia, il portale della diplomazia scientifica italiana, ma soprattutto per il mantenimento di un'identità scientifica italiana noi cittadini contiamo sulla vostra collaborazione attiva, sulle vostre intelligenze e sul vostro impegno.
Thank you. Thank you really very much to this very nice presentation, and also to Barbara Galavotti for the video that has been just presented now, that really was very, very strong in clarifying that Italian community is very close to research. We can be very proud of being among the first producer of knowledge around Europe this year for the competition of European Research Council. We were able to be the first one with 47 projects that were awarded, and that was a very important issue. but also we are also working very close with many other countries. I think in science, there are no boundaries. The science is an open activity, is a global interaction, and we need very much now more than in the past to be able to have a strong interactions among different competence, different expertise. Before we go on, I would like to ask President Villari, the president of Fondazione IDIS-Città della Scienza, to just take the floor, please.
Thank you, Gino. Good afternoon, everybody. Good afternoon to all people connected in Sweden and in Italy. Preferisco salutare in italiano il Chargé d'Affaires dell'Ambasciata d'Italia in Svezia, Mattia Mazza, il professor Mantovani di Humanitas e la professoressa Maragrazia Masucci del Karolinska Institute di Stoccolma, che ringrazio molto calorosamente per la loro partecipazione all'evento, e naturalmente il nostro chairman Gino Nicolais. First of all, I would like to thank warmly the Italian Embassy in Stockholm for collaborating with us for the organization of this beautiful event done at the occasion of the Italian Research Day in the world. As you will see in the visit later, the primary mission of Città della Scienza is to reinforce scientific citizenship among people, bridging the gap between science and society in order to bring science outside the laboratories and facilitate an open dialogue with citizens and stakeholders. This mission is implemented by Città della Scienza at local level and the national level, but also at the international level in collaboration with other science museums and science centers in the world. This is the reason why we are particularly glad to play an active role in supporting the diplomatic network for the promotion of the Italian research and innovation abroad. This action was initiated in 2018 with Italy, the Beauty of Knowledge, a traveling exhibition promoted by the Italian Ministry of Foreign Affairs that visited different continents in a worldwide tour telling the Italian excellence in the scientific field. Unfortunately, the tour was stopped by the pandemic of COVID-19, but we are already planning a new edition with the Italian Embassy in Mexico for the next September to held at Universum Science Museum in Mexico City. Hopefully today, all restrictions due to COVID-19 can be overcome by digital technologies. Let me conclude by underlining the crucial role of science communication, especially today, in facing the health emergency and supporting the ongoing vaccination campaign. So we are happy to meet you virtually today to propose this virtual tour and science talk to an unlimited audience on the net in Sweden, in Italy, and everywhere in the world. I wish a pleasant event to everybody.
Thank you very much, President Villari. Now we will have another interesting part of this meeting of this afternoon, because Città della Scienza, a few months after the spread of a pandemic, has opened a virtual exhibition to correctly inform the large public about viruses. The name is Viral Passion. We are pleased today to visit this exhibition with Silvia Bencivelli, that is a well-known science writer and a journalist. Silvia collaborates with many Italian newspapers, and she is specialized in medical and health communication for Italian radio and television. Please, Silvia, take the floor and just show us the beautiful movie that we are going to see.
Thank you, Professor Nicolais. First of all, we want to welcome you to Città della Scienza. So we have a little video to see before. So, welcome to Città della Scienza, welcome to Naples. Città della Scienza is located in Magnoli, which is a former industrial area where steel and chemical products were produced until 1990. This is why Città della Scienza represents, first of all, a model for the reconversion of the area, from the heavy industry to a knowledge-based industry. To do this, the IDIS Foundation has created a science center for the dissemination of scientific culture to the large public, but also a fab lab and a business incubator for the creation of the new job opportunities based on research and on innovation. An important event for us is the Science Festival Futuro Remoto that is organized every year to bring the world of science and research closer to people in order to present and discuss with them the benefits of science and technology in everybody's daily life. And that's what we are seeing now, and that's quite amazing. But let's go on. We are now into Corporea, the most recent building opened to the public in 2017. It is a special exhibition space, all dedicated to the human body, health topics, and the prevention of diseases. What is special here is that people can learn while experimenting science and having fun. A primary objective of the exhibition is to stimulate curiosity for scientific phenomena, especially among youngsters, and to encourage them to undertake scientific careers. Here, you can see, waiting a second, you can see the images of the museum with many visitors, as it is usual in our exhibition until this pandemic period. Publics of all ages used to visit us every day, even if our reference target is mainly families and young people, as high school students, middle schooler, and younger children. Here are the exhibition in the museum. You can see other stuff that you can see in the museum. For instance, the showcases of prostheses made with advanced materials. These are here to explain how the results of research can help people live better. And here you can see the amazing Felix robot imitating the human face of the visitors that is governed by sensor and artificial intelligence, and other funniest hands-on exhibits. This is, for example, the dancing skeleton, where you can test and understand how your musculoskeletal system works. Then we also have a funny bed of nails. Here it is. The bed of nails, in the bed of nails, is it possible to lie down as fakirs do, challenging the gravity laws and understanding why we do not get hurt. Or I hope so. Here we have another section. We are ready to see that. This section is dedicated to the heart and to the circulatory system. And we see another amusing exhibit where visitors can test their heartbeat and observe its frequency looking at these jumping balls. So the Science Center is a place of exploration for everybody, and also a place to share the understanding of scientific phenomena and to share that with friends, with parents, but also with researchers and the expert. And this is the Planetarium. The Planetarium is a large hemispherical room where you can experience 3D movies and very nice simulations of the sky in the night. And you can understand more about astronomy and astrophysics. And that's marvelous, as you see. Città della Scienza is definitely an amazing place where you can have good time with family and friends, overlooking the beautiful Naples Gulf with a view of Ischia and Procida Island on the background. So we really look forward to welcome you soon. And now I think we can start the visit in the exhibition. So we are going to talk about viruses, because today we feel like we are expert on viruses, but in reality, viruses are still not very much known. And anyway, its definition is also still very complicated. The one that I prefer is a bad news wrapped by a protein. The one who invented this definition, this funny definition, was a Nobel Prize, Peter Medawar, as we can see there. He was an interesting historical character, by the way, and he won the prize for the study of immunology. But you don't have to take this definition as a very much scientific one. It's much ironic, maybe. But we have to think that viruses could be sometimes for us bad news. A virus per se is not bad and is not good. It's just a piece of genetic material wrapped by a protein. So genetic material, so a news in that sense, Medawar was right, but neither bad nor good. Then, if you want, for us a virus is an intellectual challenge, an object of this kind. Is he alive? So virus as an identity, as an information, a genetic material, can replicate, but cannot do it autonomously. It is a parasite. So with viruses, we are on the border between life and non-life, between biology and biochemistry. That's why another definition, very effective to overcome this impasse with irony, is borrowed life. And then viruses are really tiny. We discovered them at the end of the 19th century, and we called like that viruses, using a Latin word that means poison, because we identified that there was pathogenic agents smaller than bacteria, but we realized that the nature was uncertain. Then we realized that there was a certain disease of tobacco plants, that it was contagious, but it was not caused by bacteria. It was a Netherlandish chemist, Martinus Wilhelm Beijering, who did this discovery. This is Martinus. But then he was not able to see that virus. It was too early, because until the 30s, the viruses weren't easy to study. We were able to have them grow in culture. We were able to understand that they infect plants, animals, bacteria, humans being, and then we were able to produce vaccines. But then we didn't saw them until the 30s, when we discovered the electronic microscope. In the 50s, we understood that viruses are genetic stuff enveloped by a protein shell, and we understood that they get in host cells, and that these host cells may be the one of our nose. But then, look at the picture that we have here. Look at that. We have these two pictures with two families, 1 century apart. They all have the faces covered by masks. These two pictures represent two biggest viral epidemics that hit us during 1 century. Is it impossible to talk about viruses without talking about epidemics? And we will also do during this visit. Why do the epidemics appear? Because viruses transform, move, they pass from one species to another one, doing a spillover, and when they reach our species, we say that they appear, and we call them emergent. Sometimes these viruses causes illnesses in us, and we find ourselves without our immune system ready to fight that virus, so we get sick, and we may often get sick altogether, and this is an epidemic. The worst epidemic have been caused by the viruses that we are going to see in the next room, which is the room dedicated to the RNA viruses. So let's go to the next room, RNA viruses. This is something like a room about history, not just about virology, because each of these viruses deserves a reflection per se. So let's see, for instance, the Rabbi virus. Rabbi is a very, very ancient disease. The first time it was talked about Rabbi, it was in the Hammurabi Code. I told that we were able to produce vaccines against viral diseases before having seen them. And that has happened to Rabbi. the vaccine has been developed by Robert Koch in 1885, but we see the virus for the first time at the electron microscope in 1962, so almost 1 century later. In reality, at the beginning of the last century, a young Italian virologist identified inside the neurons of animals that were infected some cytoplasmatic inclusions, and he associated them correctly to the disease. The name of this young Italian researcher was Adelchi Negri. He was, here he is. He was a student of Camillo Golgi, who have been the first Italian Nobel Prize, Medicine Nobel Prize in 1906. And Adelchi Negri, he died very young for tuberculosis. In reality, Negri was not able to understand that it was a virus. He understood that it was binding to the illness, but not that it was a virus. And then it was another Italian virologist, Alfonso di Vestea, that later did it, understood that. And in the same time, we have to admit that there was a French one. Notice that it was the very first years of the 20th century and that we just began to talk about viruses. But let's come back to the center of the room. And then we have to see that's here, the hepatitis C, the C hepatitis virus. This is one of the new viruses in our history. Its existence was hypothesized in the 70s, following the onset of the hepatitis from transfusions. But for a long time, it was called virus non-A, non-B, because we couldn't find it. Only in the 1988, it was identified, and we called it the virus of hepatitis C hepatitis. This virus can cause chronic disease and even lead to the developing of cancer. Indeed, it is the main cause of primary liver cancer. A vaccine for it is still missing, but we have a cure. A cure existed for some years, so therefore we hope that the virus can be eradicated. We hope to block the chain of contagion thanks to the therapy. For the discovery of this virus, a Nobel Prize was given in 2020, and it has been an act of symbolic value because we were in the middle of the pandemic, this pandemic, which is right now in act. So come back to the center of the room because I want to show you another interesting virus, which is the polio one. Here we also have a very beautiful history, especially linked to the development of vaccines. This vaccine almost permitted to have disappeared from Earth this disease. It was a devastating disease. It was a very, very severe illness in children that was causing epidemics. The last epidemics we have had in Italy was in 1958. The Ministry of Health was just born. And we started a vaccine campaign that had some difficulties at the beginning. So we were talking, as we are doing right now, about which vaccine and mandatory or not, and so on. But the history of polio virus and its vaccine is probably the most successful story in the history of science, along with that of smallpox. We will talk about that later. So come back to the center of the room, because next to these successful stories, there are other that are not that nice. Other story linked to the epidemics and to this kind of viruses. So let's see the measles virus. Measles, this virus was born many centuries ago due to the mutation of the bovine pest virus. And it appeared when we began to domesticate cattle. It could be that the Athens pest in 430 before Christ, the one who was described by Thucydide, was measles. We tend to underestimate the severity of this illness. We have a vaccine since the 60s, but this virus is very, very contagious. And we still are unable to vaccinate enough people to be able to reach herd immunity. And so periodically there are new epidemics. 2 years ago in New York, starting from a religious community that was hesitant, that is still hesitant for vaccination, a measles epidemic has started, and it has been interrupted just thanks quarantine and widely spread vaccination. In the same times, there was an epidemic in Congo. So come back to the center of the room, because Congo brings us to another virus, which is this one, another RNA virus, which is Ebola virus. Ebola virus is born in Congo, as I said. It has been identified in 1967 close to the Ebola River. It has a high mortality rate, and times it causes epidemics. There is one happening right now in Guinea. These epidemics have always been localized in Africa. And unfortunately, this explains the limited attention that we dedicated to this virus. From 2015, there is a vaccine, but at this moment, it's available only in very limited quantities. But it's important to say that this vaccine was a prototype for the construction of some of the anti-COVID vaccine that we are using right now in Europe, the ones made from a viral vector. So it's important a lot also for us. Come back to the center of the room, because I want to show you other two RNA virus. The next is HIV virus. This is also a very recent virus in our history that emerged officially in the 80s. It was a big shock for us Europeans, because we thought we were protected somehow from diseases, even more from the viral ones that we considered light and under control. But HIV causes a very severe disease. Unfortunately, even if nowadays we have effective therapies, we must say that there is no vaccine yet for HIV. This is a very complicated virus. It's different from the other ones that we find in this room. Technically, it is a retrovirus, so a RNA virus, but once it enters in the host cell, it transforms its RNA in DNA, and it can integrate its DNA into the one of the host cell. And it's a virus that mutates very, very fast, and moreover, the host cell are that of the immune system of us, as human beings. That's why this virus is a real challenge for the production of vaccines, and there is still very much to do for prevention. Come back to the center of the room. The next virus I want to show you is a very important one, and is a flu virus. The flu virus, which is the virus that we exchange every year with other animal species. And it's always a little bit different compared to the one of the previous years. So it changed year by year. The flu virus during the 19th century caused many pandemics, some more severe than others, and every year, in any case, put millions of people to bed. We have had a pandemic in 1958, then in 1968, for 40 years nothing, and then the swine flu in 2009. But mostly, and then most of all, and now we go back to those people in the picture with the mask on the face that we saw at the beginning of this tour. Spanish flu pandemics was the most important one that lasted from 1980 to 1920. Spanish flu, from what we know, caused 50 or 100 million deaths of a world population of about 2 billion, and half billions of people got sick. We call it Spanish flu because during the First World War, Spain was neutral, so there was not the military censorship, and the news circulate freely. Also the news about the disease of the king, and in this case was the King Alfonso XIII. This gave the wrong impression that Spain was the country with the highest incident rate of the disease. And it was wrong, but we call the flu in that way. In reality, very likely the virus appeared with a spillover that happened in the US, and then it moved with the military corps during the war. Many people died, such as Guillaume Apollinaire, the poet, Egon Schiele, Gustav Klimt, the painter, Max Weber, the sociologist. Even Frederick Trump died, the grandfather of the more famous Donald Trump. No corner on Earth was saved. No Europe, America, Asia, even the Pole Island on Pacific. Spanish flu was so deadly also because of the war and of the hunger that made the population weak for the lack of antibiotic, but mostly for the fact that we didn't know the cause of the disease. This is why the adopted measures were sometimes a little bit fanciful, sometimes magic. But in the cities, we have took some measures of public hygiene that were similar to the ones we are adopting for the pandemic that is happening right now, like the shutdown of very crowded places, messes, local parties, mask, disinfection, and so on. And Benito Mussolini, who at the time was a journalist, on Popolo d'Italia, was a newspaper, wrote that we should have took it out on the filthy habits of the handshake. We should have give up to that. Our country was hit hard. The estimate were around 375 and 650,000 dead out of about 38 million of inhabitants. Why estimates and why so wide? Because there was a war, and it's impossible to count precisely the amount of people that were affected and that died. Then there are coronavirus. Excuse me a while, I'm having hard difficulties taking time. Okay. Coronavirus, this is the family to which belongs also the infective agent that causes COVID-19. Coronavirus are very spread. Generally, they only cause cold and very light diseases. But we need to remember that in the first years of 2000, they already caused 2 epidemics. They happened only in Asiatic countries, and they were contained somehow. One was SARS in 2003, and the other one was MERS in 2012. They were both very deadly, but not that much contagious. So we already had some alarm signals about the possibility of the advent of a new coronavirus. And let's see, the coronavirus is the one that is causing the actual pandemic, which is also the reason that we are online right now, and the reason that we are explaining this kind of stuff online with this kind of exhibition. The spillover that led to the arrival of this new virus happened probably in the autumn 19 in China. And as we saw, spillover, which is a jump between species, is a natural fact that happens continuously on our planet. This time, it surprised us because it was a century that in Europe we didn't have severe respiratory pandemics, or at least 50 years if you consider the pandemic of the 1968. But we have to say that the commitment of the scientific community was enormous from the beginning. Today we have hundreds of thousands of papers published on the theme, and the vaccine, first time in history, that was ready in less than 1 year from sequencing of the virus genome. So this virus will change us. It has already changed us. We have online, we do everything online, but it also changed science. Today there are much sharing between scientists. There is more discussion, more participation, even outside the scientific community. There is a lot of attention on communication, including the one to the public, and a higher request of education. And this may be a good news. So here in this room, you can find material, in-depth material. And let's go to the next room, which is the DNA virus room. Here we are. No, here we are. We are going. Okay, here we are. So viruses in this room are the cause of very common diseases and generally not very severe. But for many of them, it was discovered that they can cause tumors. So things changed a little bit. Virus can cause tumors. We know that for a century. The discovery was awarded, also this discovery was awarded with a Nobel Prize in the 1966, even if it was half of a century that we understood that. But the discovery happened at the beginning of the 20th century, so long before we were able to see viruses, and it happened with a chicken virus. Hepatitis B virus that we are seeing here, and then other viruses, papilloma virus and so on, they are related with liver, stomach tumors and head neck tumors, and then lymphoma too. Recent study on these tumors discovered traces of viruses in 30% of cases, and WHO calculates that almost 15% of global tumors are actually caused by viruses. So don't underestimate that. Some of these viruses are viruses that never leave us, like herpes viruses. Once we catch one of these, it remains hidden forever in our body. For instance, the cold sore virus remains in the trigeminal nerve and reappears whenever our immune system is under stress. But having talked so far about epidemics, I would like to conclude the visit with a story that is meant to be a wish for everyone. We mentioned it 2 rooms ago. Here is the smallpox virus. Okay, the smallpox virus. The smallpox was a very severe disease, but it no longer exists. Those under the age of 45, perhaps 43, in Italy did not even have to get vaccinated against the smallpox, because in Italy, all over the world, because the disease completely disappeared from the face of the earth, thanks to the fact that previous generations have been vaccinated. We people of my age and our children, we had inherited a world without smallpox, without this virus that science began to fight against centuries ago. For us Europeans, the story began in 18th century, and as a key date, May 1796, the moment when the first vaccine in history was tested. The story begins with a doctor, Edward Jenner, and a child called James, and a milker, Sarah. Sarah had tracked the form of the disease. According to the popular wisdom, she would not have fallen ill with a human one. So Dr. Jenner transferred Sarah's disease to James, and James officially became the first vaccinated child in history. Since then, the smallpox has been made available. The smallpox vaccine has been made available to many other children, and thanks to the generalized vaccination that have been going on for decades in the last century, the smallpox virus has now disappeared. Last case was in 1977, and since 1980, it has been declared extinct. So 40 years have passed. Since then, only one other viral disease has been eradicated from the face of the earth. This is the bovine pest. And yes, this is the one from which measles have derived. So thanks to vaccines and thanks to complex health strategy, public health strategy, we could get rid of it. We could get rid of measles, but even before we could get rid of polio, and sooner or later from other diseases. Will we ever get rid from SARS-CoV-2? No, for the moment it's not very likely, but we are learning how to defend ourselves, and we are learning it day by day. And for that, we have an incredible, powerful weapon, which is scientific research. And so I will like to thank every scientific researcher in the world, and mostly the Italian one, because today is their party. And with that, I give the floor back to Professor Nicolais.
This virtual exhibition and really is very important for many people. They don't really are aware of this kind of viruses. Now I like to introduce for this Italian Research Day and scientific collaboration between Italy and Sweden two really first-class scientists, both Italians, but one working in Italy, another working in Sweden. I think this is very interesting to see, because we very often we think that in Italy there is not too much science that is produced, but that's not true. And very often also we think that everything comes from abroad. But when we go in deep, we see that this science that has been produced abroad is coming from Italian scientists. And this is the case today. First of all, we will have Alberto Mantovani, good friend of mine since many years, and is, among many other things, also a most cited scientist working in Italy, and this is a very a pleasure for us. And on the other side, we will have Professor Masucci. She works in Karolinska Institute, and I'm glad to say that I've been there to teach some course many years ago, working in totally different fields. That was rheology of pulp paper. That's you know that in Sweden paper is an important issue, and I was just a guest professor working there for some time, and we had a very strong collaboration with Sweden, and we developed also many nice research papers, but also patents. And that's really, I'm very glad to see you tonight. So I will leave the floor to these two scientists, and we will start with Alberto. And Alberto has many recognitions in many cases, but at the moment he's working with in Milano, and is a professor of pathology and scientific director of the Clinical Institute Humanitas, that everyone, not only in Italy, knows for the excellence, not only in medical activity, but also in science. Please, Alberto.
Thank you, Luigi. It's a pleasure to be with you, and it's particularly a pleasure to be together with my ragazzi Masucci. And I would say that I'm at home in Scandinavia because I'm an honorary member of the Scandinavian Society of Immunology. I'm very proud of that. So I'm at home. I belong to the two words, Italy and Scandinavia, and Sweden in particular. And one of my last trainees is now in Sweden doing. Andrea Ponzetta doing excellent work. So let me try the most difficult part of my afternoon, that is to share my slides. I hope, share screen, I hope I can make it. Yes, here it is. Can you see it?
Yes.
Can you see my slides? Yes, good. And can you see it full screen now?
No.
Yes.
No, it's not full screen. We'll see the side view also.
Oh, gosh.
Try to switch.
Yeah, is it moving now? No. Sorry, I knew it was going to happen. So let's go back and I'll try again.
Okay.
Yeah.
Is it working now?
Yeah.
Yes.
Okay. So Silvia Bencivelli already mentioned the pest of Athens and Thucydides and the measles-like disease, probably, and that is probably, to the best of our knowledge, the first sensing, the first perception of immunological memory, which is at the basis of vaccines. And because Thucydides described that the people who had this disease, this measles-like disease, and who did not die, were immune, did not get the disease a second time, or got it in an attenuated form. So I'm an immunologist interested in innate immunity, and in particular, tumor-associated macrophages. But 1 year ago, I was confronted with this virus and with the emergency related to it. This is actually the representation of the virus by 3 of my 8 grandchildren. And you can clearly see here the spike protein. It's not as fancy as the image.
You're actually not moving forward now. Mm.
Oh, gosh. Am I moving now?
No.
Am I moving?
Okay.
Is it moving now?
Yes. Yeah.
Okay. So this is the representation by my 3 grandchildren. And you can see the spike protein here. So this when confronted with this new enemy, and we were very badly hit 1 year ago in northern Italy, I went back to my our Roman Greek roots, and the perception that I know that I don't know, Socrates, and this being the premise for making progress. And this is a summary of the challenge and progress we made, the virus, and I think that Maria Grazia is going to discuss the variants. We realized immediately that and we all know that not everybody gets sick, not everybody gets seriously sick, and the first line of resistance, innate immunity, the adaptive immunity, the directors of the immunological orchestra, the T cells, then the B cells that make antibodies. And I want to remind everybody of the seminal work done in Sweden by Hans Gustav Lundgren, a friend, and other people who have shown that people who have negative for antibodies do have, asymptomatic people do have T cells that respond to the virus. And this was really, I think, a seminal observation and discovery. And of course, the outcome of the interaction with the virus is determined by viral load. We are going towards summertime, gender. Ladies are much stronger than we are, and that is no discovery for me in life in general. Aging, general paradigm of inflammation formulated in our country by people like Andrea Cosarizza and others, lifestyle, obesity, and genetics. So immediately we had a question mark. I mean, we see an apparent much greater lethality in northern Italy compared to China. Is it related to genetics? So in our institution, and Stefano Duca and Rosanna Selta, we started genetic analysis. I think this is still the first genetic paper. We had it in open access much earlier than immediately. And then we were part of a European effort that involved many countries, a genome-wide association study. And this is the Manhattan plot, the chromosomes, and there is clearly a skyscraper on chromosome 3, and there are chemokines and chemokine receptors, and I've been involved in some of the early description and identification of chemokines. So this has to do with innate immunity and inflammation, blood groups, a question mark for a while, and there is more data now. So there is a genetic components in the severity of the disease of COVID-19 disease. And collaboration is key, and open access to data is key. And this is it's impressive for me. And this is the human genetic architecture of COVID-19. It's in open access now. It's coordinated in Finland. And we are part of this worldwide effort. And this paper, which is now submitted, it includes 2,800 authors. And so it's amazing change. But this emphasizes the importance of no boundaries, as Luigi emphasized, and collaboration. So when innate immunity fails, I'll get back to that. Let me remind you that innate immunity and cells that are not mentioned here, NK cells, which were discovered in Scandinavia, in Sweden, and at Karolinska. And when innate immunity fails, when adaptive immunity fails, you have uncontrolled inflammation. You have new diseases, which is interesting. This is MIS-C, an inflammatory disorder, systemic inflammation, which affects children. Actually, it's pre-adolescent or adolescent, quote, children. And then long-term sequelae. And we used to call it long COVID. The official name is now PASC, so new diseases. And the first intervention, therapeutic intervention that affected mortalities are glucocorticoid hormones, low cost. The importance of a national healthcare system, the study by Horby et al. In UK, and Maurizio Cecconi in our institute, he is the head of intensive care unit, was part of the crystallization of guidelines by WHO, and part of clinical studies. Actually happened to be, again, the contribution of the country, one of the 3 heroes of the pandemic, as named by Journal of the American Medical Association, JAMA. And I should say and emphasize that we are concerned now, because this effort, and again, this is the recovery trial, showed that glucocorticoids are only active in a well-defined window. And we are concerned because in our experience, in our emergency care, we see patients treated early in the disease with glucocorticoids. There is no evidence of benefit, some hints of benefit, and we have published this concern of ours in a paper, as we do usually. Well, always, as we do always. First scientific paper, and then we talk. So let me go back to the innate immunity, just to emphasize that the landscape, our perception, our understanding of the disease has changed. The disease is at the intersection of genetic predisposition. I emphasized the work, the collaborative work worldwide now, and the original work that we did. Genetic immunodeficiency, which is unmasked by the virus, and this is work that was done by an expatriate, by Jean-Laurent Casanova and Gigi Notarangelo at NIH, and autoimmunity. Autoimmunity triggered by the virus, and uncontrolled inflammation. This results in uncontrolled inflammation. And we are very interested in uncontrolled inflammation and inflammation in general. For instance, this is a study, and I emphasize this in terms of method. We started off by doing RNA-seq in our own patients, in the monocytes of our own patients, cells of the innate immune system. Then we moved to single-cell analysis, and found that this gene of ours that we cloned many years ago, a distant relative of C-reactive protein, was very high and expressed in myeloid cells. Then we did single-cell RNA-seq in the lungs, and we also took advantage of access in an open world of patients in United States and Israel, and then back to autopsy sample, and then impact on survival. And this happens to be a very strong, bad prognostic indicator with hazard ratio around 20. And again, collaboration within the country and outside of the country. Vaccines, in the last 5 minutes, I want to very briefly make a comment on vaccines. And I want to remind you of a forgotten anniversary. This is the 300-year anniversary of the first clinical trial on a, let's call it vaccination strategy. Actually, it's an inoculation, immunization strategy, using smallpox, real smallpox, to immunize. And the pioneer here is this lady, all too frequently forgotten, Lady Montagu, and she had immunized with this strategy her own offspring. And the trial was run in the Newgate prison. It also happens to be the 300-year anniversary of the first real-world assessment run by Reverend Cotton Mather in Boston, showing that actually the immunization strategy did work in real world and not only in the prison. So vaccines, well, we're all familiar with the platforms, classical all bug platform, classical protein platform, the messenger RNA platforms, we all know that. I want to emphasize, this is the adenovirus platform. I want to emphasize that this platform capitalized on the work done by an Italian who passed, namely Riccardo Cortese. Riccardo, I think, was really the pioneer who built the foundations for exploiting adenoviruses in vaccines. We need more. We need many vaccines. And of course, one of the challenges is using different platforms, possibly complementary platforms, because of the conditions, the actual conditions, and because we may get the best of different worlds. For instance, a viral platform and a protein platform or a messenger RNA platform. And this is one of the challenges we have. I want to make, just briefly mentioned, a story, our own story here, single dose vaccination. These are subjects who had experienced COVID. And this is in press, but as usual, we put it in open access. Maria Rescigno coordinated the study. We found that in ex-COVID patients who had clinical COVID, one shot of the mRNA vaccine was plenty. And I think we were together with Florian Kramer, the first to report this in open access. Many groups had the same observation. And the impact of research, this has resulted in statements by I'm aware of Spain, France, Italy, Germany, that people who had experienced COVID-19 should only get one shot of the mRNA vaccine. And at the time of vaccine shortage, this means potentially to save 2 million doses of vaccines in Italy and worldwide over 100 million doses. And there is a statement by the various groups, again, collaboration worldwide, a statement by the various groups who contributed, this is our own paper, pointing to the need to have this as a procedure for the safety of the individual and for society. Vaccines, or some vaccines, for instance, polio was mentioned, have an impact, what has been called pathogen agnostic protection. Namely, they confer protection that goes beyond the pathogen to which they were designed. Mm-hmm. And we did I was involved in some of the early prehistoric studies on innate immunity in the field and macrophage activation. And Mihai Netea, colleague and friend in the Netherlands, did much of the work, mechanistic work on epigenetic remodeling underlying what he, and this is his term, trained immunity. And this is a question we don't know whether the current COVID-19 vaccines train the innate immune system and whether this is relevant to resistance, for instance, against different variants. And finally, a quick look at global health. We have, I've been part for 5 years, served in the board of the Global Alliance for Vaccines and Immunization. And Gavi is not only a good Italian wine, it is also a global health initiative which was born to fight against the 2.5 million of children who used to die at the time every year because they didn't have access to the most elementary vaccine. Now the figure is 1.5 more or less, so it has improved. And now Gavi is at the main driver of the COVAX initiative, whose objective is equitable sharing of vaccines. And I do feel, and we do feel, this is Rino Rappuoli, the president of the Italian Society of Immunology, Angelo Santoni, and myself, we do feel that vaccines represent an achievement of civilization, a human right for each newborn on the planet, and a health insurance, a safety belt for humanity. And COVID-19 has vindicated this statement, and we should not forget equitable sharing of vaccine. And I mentioned, and Silvia mentioned, the history, and if you are interested in contamination between literature, history of science, and immunology, well, Piccolo Teatro di Milano and the Fondazione Humanitas per la Ricerca made an effort on this line, and here is the link. And I think that this is an Italian painter, Lucio Fontana. I think that the challenge for us are these cuts, to go through the cuts with science at the service of global health. And thank you for the privilege of being in Scandinavia with my heart today, and in Sweden in particular.
La voce.
I will just now leave the floor to Maria Grazia Masucci, and we will be just opening for some question later on, after your speech. Thank you, Maria Grazia.
Okay, let me try to see whether I can be as good as Alberto in this. Share screen, application window, this, share, and let's see, slideshow. Let's see. What do you see? Is that okay? You see nothing?
Nothing at the moment.
Oh my goodness. Now?
Yes, we see something.
Okay. Okay. Let me then start from the first slide. Okay. First of all, it's difficult to come after such a beautiful presentation by Alberto, but I thought that since he so beautifully described or started to put into context what we do against the SARS-CoV-2 that caused the disease, we should also take a step back and look at the enemy itself, at the virus. And picking on what Silvia already said, I am going to try to give a little presentation of the virus and what are the questions which one tries to ask, or which one is which we are globally asking, in order to understand how this virus can cause disease. And from this knowledge, we hope to obtain information on what to do, which kind of measures, medicine, treatment, and strategies of combination of treatment we should use in order to combat the disease. So, as Silvia has already pointed out, SARS-CoV-2 is an RNA virus. And this is a little picture, as you can see, an electron microscope picture simplified as to what is it done. It contains a little piece of RNA, the genome of this virus and RNA. And since the nucleic acids are very fragile, this is protected by the protein capsule, where a little box made up of protein, which made this resistant. And then the protein again is enveloped into a membrane, which the virus steals from the cellular from which it is produced. And on this membrane are attached or are inserted all these little spikes that look out and give us these corona-like structures. And this is what the virus uses to bind to receptors on the cells that it will infect. As was already mentioned, there are many coronavirus in the environment, and most of them are animal viruses. In fact, the reservoir of these viruses is bats. And the reservoir means that this virus can infect bats without causing any disease. So it can replicate in this virus, and there are in this type of animals, and there are at least 2,000 or 3,000 different types of coronaviruses in bats. There are also coronaviruses in all mammals, and one of the big, it is in fact a big economical burden, because epidemics of coronavirus can have a very destructive effect. For example, an epidemic of swine or pig coronavirus in the United States almost wiped out the pig industry in the end of the last century. The first human coronavirus was actually discovered in the 60s. And it was discovered as a virus that is a relatively benign virus. It causes approximately 20% of all common cold in the winter. And one interesting thing to point out is that since this virus does not promote the development of a persistent immunity, as Alberto has already mentioned, basically every year, and the immunity to this type of coronavirus persists for approximately 10 to 12 months. Every year we are susceptible to reinfection, which is a really thing to keep in mind when we think about the new virus. Since this 1960, there have been 3 serious emergency of new coronavirus in human, which are probably and are most likely due to a cross species. So the virus suddenly managed to jump from an animal to humans. And these have already been mentioned. The Sars epidemics that started in southern China. It was a relatively, seen in our today perspective, it was really a small thing, only 8,000 cases. But it was quite deadly, 11% of cases, 11% of mortality. For some reason that we still don't understand, the epidemic lasted between 2002 and 2003, and then the virus completely disappeared. A second variant of human coronavirus appeared in the Middle East in the 2012. And these epidemics of what we call now Middle East coronavirus infection is still ongoing. This is a very deadly virus, 34% of the cases, 34% mortality. But the number of cases is very limited. And then we come to what happened suddenly in 2019, where another coronavirus crossed species and came to humans. And well, as you can see, the difference between the number of infected cases is staggering. 8,000, 3,000, 140 million. So this new virus is extremely more infectious of the first compared to the first two, but it's also less deadly. At the end of the day, it is a significantly bigger problem for humanity than the first two. So one question which is, of course, of great importance and which has been debated a lot is the origin of SARS-CoV-2, of this new pandemics that we are now suffering through. And our analysis of this is based basically on epidemiology, but mostly on sequencing, because by looking at the genome, we can see how the genome changed, and we can look at different similarities, and we go back to this later. So the SARS coronavirus, we know one thing for sure, it comes from bats, because the genome of the new virus, which infects humans, is very similar to the genome of one of the genomes which is found in bats. But it is not identical, and it has, in fact, additional similarities to viruses which are found in pangolins. Some of the important characteristic of this virus in terms of infectivity, in fact, appears to come from pangolins, which suggests that the new virus, which is often the case when a virus jumps a species, goes from one species to an intermediate species when some selection is done, to the final species when most infection is, where the highest level of infectivity is achieved. And this new virus, which now infects us, has two important changes compared to the bats and pangolin virus. This virus has the important changes confer to this virus a selective advantage in that this virus binds much more efficiently to its cellular receptor, so the portal entries that the virus uses to enter into the infected cells. And also the mutation, these changes change the glycosylation of the virus, which means that the virus can in a way cover itself and hide itself from the host immune response. Another issue which has been extensively debated is when did this occur, and when did the jump of species occur, and maybe we will never have final answers to that, but based to the analysis of sequences, this is likely, the jump of species is likely to have occurred early during the autumn of 2019. So most certainly the virus did not emerge in the Wuhan fish market or wild animal market, but while this, or the market, was probably the first case of super spreading events, and now we are familiar with the word of super spreading events, but most likely the virus slowly adapted to humans over several months before this high cluster of cases appeared. Sequencing is what tells us about the virus. And sequencing is also what tells us about the evolution of the virus. And this is an RNA virus. And the RNA viruses have one characteristic, as I said, that their nucleic acid is an RNA. And in order to replicate this nucleic acid, to make copies of it, one has to use a specific enzyme which uses RNA as a template for producing a new RNA. And these enzymes are usually very imprecise. They make a lot of mistakes when they work. So basically, every time the virus replicates, there will be mutations which occur in the virus. And in fact, if one goes back and look at the first sequences that already our Chinese colleagues put in the public domain, and which are and the collection is rapidly increases, the first species, they had 99.9 identity with the first sequence. So for a month or so, 99.9, 99.8. This virus contains 30,000 base pair. 99% identity means one changed base pair per virus, which means that every one of these viruses, which was sequenced already from the beginning, contain at least 30 differences compared to the original virus. So most of these changes are, however, completely irrelevant for the virus, or actually beneficial, because they occur in a place where this virus, they will stop producing proteins, and therefore the virus will not be infectious anymore. Or they will occur in a place where they will not change the sequence of the proteins which are produced, so the virus will be functionally identical as the previous virus. But sometimes, and this is why sometimes these changes occur in places where the modification which are induced actually enhance the capacity of the virus to infect, which we call fitness, or replicate and infect, which we call fitness. And this is what makes this is a selective advantage, because these new viruses, which are generated randomly, will be more effective when it infects the next host, will elicit, or where the immune responses will not be as affected, so there will be more virus produced, etc., etc., etc. And this is what today, more or less 1 year after the first appearance of the virus, we start to appreciate as the variants. The variants, they would carry specific mutation in places that make them more fit to infection. More fit to infection does not necessarily all the time means more deadly. It could be, as we start to seem to observe, and there are reports coming from that, is more fitness makes them more easily infecting, but maybe in some cases this is maybe less serious, or it may take different characteristic. As it seems, there are reports suggesting that there are variants emerging that will give a different clinical picture as compared to the original virus. Some few more words on the virus itself. So in order to combat this enemy, we have to, what do? We have to understand how it actually works. And this is where all this sequencing gives us a start off. And this is where the analysis of what the virus, which are the protein encoded by the virus, becomes extremely important, because we have to understand how the virus functions eventually. So as I just mentioned, this is a big virus. I don't know, can I use the do you see this? Do you see my pointer?
I see, yes. Yes.
Okay, very good.
Not the pointer, okay. Not the pointer, but I see the slide, not the pointer.
Okay, you don't see the pointer. Ah, that's a pity. Okay, well, okay.
You can use a laser pointer probably by clicking the right.
Maybe this is the laser pointer. Can you see this now? No. Okay, maybe we don't try to be too technical, otherwise it will be too complicated. So this virus, so I said, the 30,000 base pairs is the largest human RNA virus known, by the way. And it produces 3 types of protein. It produces proteins which are non-structural and which are important for the virus to replicate. There are important proteins which are structural, and they are important for building the virus. And these are the red proteins here. And there are a group of proteins which are green in this picture, and which are proteins that the virus uses to modify the cells in order to make it to sort of capture all the function of the cells for its own replication. So the non-structural protein include the polymerase. I mentioned that this virus needs its own enzyme for replicating. It's an RNA-dependent RNA polymerase, and the 2 proteases. And the proteases are very typical for RNA viruses, because the viruses, they start their life by producing a very long protein. This long blue thing is just one protein, which has to be cut down into the individual components in order to make all the functional protein of the virus. So the proteases are really important, and the polymerases are really important, because these are proteins that are not contained in our cells. We do not have this protein, and therefore we can hope, by understanding exactly how this protein works, to use those as target for intervention for the production of antivirals. Then there are the capsid proteins, which we have described, the one which protect the virus. And then this is a very important spike protein, this S protein, which is or at least the most important structure, the structure that the virus uses to attach to the cells that it will infect. And this knowledge help us to get a deeper understanding, let's say, or of what we need to understand better is what is the life cycle of the virus. How does the virus achieve infection? Because if we could block this pathway at any step, we would of course have a way how to inhibit infection itself, prevent disease, etc., etc. So we know that the virus, this little thing with all the spikes on the side, attaches to the cell membrane through this receptor, which is there in red. And then we don't know what happens next. There are two possible ways. Either this will result in a receptor-mediated endocytosis, where the virus enters the cells into a little vesicle, or this will reside in a direct fusion of the virus to the cell membrane and release of the viral nucleic acid into the cytoplasm. Also, if the virus enters through this pathway of the small vesicle, somehow it will have to, the vesicle will have to break and liberate the nucleic acid. And this will start with the help of cellular function, the production of the viral protein, that through the viral protease, it will result in the production of the big enzyme, the viral replicase, that will produce new virus and will produce also the mRNA for the production of all the viral proteins. And this is a particularly sensitive step for the virus, because this replication will involve the production of specific mRNAs for each one of the viral proteins. And at this stage, there will be a recombination process which occurs. And this recombination process and the proteins, viral proteins that regulate this recombination process are again important target, because it is during this recombination process that the jumps between species may occur. When two different viruses infect from different species happen to enter into the same cells, the recombination between two different viruses will create a completely new virus, which has important different characteristics. And then the next step will be for the virus to reassemble itself and to be released as a new infectious virus. And if I had to now mention what are the important things which we want to understand here, we go back, is the entry point is a very sensitive point. How can we block it, and what are the important functions that are involved in this step? The proteases, unique proteins for the virus. If we would block them, we would prevent the production of the viral replicase complex and therefore completely block virus replication. The replicase itself, this is an RNA-dependent RNA polymerases. Many RNA viruses have them. Some inhibitors of these enzymes have been very successful, for example, for hepatitis C. We do not have yet, and one is even tried to use an inhibitors for the Ebola RNA polymerase to try to use for this virus. It doesn't really work very well, but it is definitely a very important target. The recombination step will be again a very important step for inhibition. And even if we know very, very little about this yet, this assembly and exit pathway is also a potential target, because if we can inhibit the exit of the virus, we will inhibit virus replication. Oh, I can see now that I have that summarized in this slide. So for inhibiting the first step, the entry of the virus, what we may think about is neutralizing antibodies. And there is a lot of discussion going on neutralizing antibodies and how they could be used, but basically they would inhibit the binding of the virus to the receptors. There are important caveats in the use of this type of strategy. A very similar strategy is to use something that resembles the receptor on the cells, and this will again prevent the binding. And the third strategy would be to inhibit those cellular, in this case, enzyme, like this one with this impossible name, TMPPP, that is required for the spike protein to actually start the fusion. And whether this occurs at the cell membrane or whether this occurs in this vesicle, in the endosomes, we still don't know. Chloroquine, for example, which has been discussed very, very intensively, could be one of the enzymes that inhibits this step, but it in fact turned out to be very ineffective, but there are possibly other enzymes. And we have talked about the inhibitors of the proteases, the inhibitor of the replicases, the inhibitor of the recombination, and more inhibitors are potentially on the way. And I don't know, I unfortunately don't see my time, so I don't know how much time I have and how much time I've used, but
5 minutes.
I have 5 minutes. 5 minutes will be enough. Because I want to say very, very, very briefly about, we have talked about the virus, we have talked about it works, how it works, and the questions which we are asking as to how can we interfere. But then the real question, and Alberto already touched on this, is how does it cause disease? What are the important pathogenetic event in this virus that makes it different from other viruses? And there are two things which are emerging in this area. One is the use of this particular receptor, the ACE2 receptor. This is an angiotensin converting enzyme, and this is an important enzyme physiologically in the cells, because this enzyme in the cells actually is participates in 3 important signaling pathways. On one pathway, it actually processes angiotensin 1 and angiotensin 2 products, and has basically a very strong inhibitory activity, or inhibits the activity of these 2 enzymes, which are regulating blood pressure, blood permeability, and inflammation. On the other side, it inhibits the bradykinin pathway, and bradykinin 9 is a strong pro-inflammatory product. So ACE2 breaks down BDR9, breaks down angiotensin 1 and into this angiotensin 1,9, which is an inhibitor, and or angiotensin 1,7, which is an inhibitor. When the virus infects cells, it binds to these receptors. It promotes the and it promotes the degradation of the receptor. So basically, this pathway, which usually are inhibited, which pro-inflammatory pathway, which are inhibited by ACE2, suddenly can be activated. And this is likely to be a very important component of the type of pathology which we observe, with defect in cardiovascular function, coagulation, blood pressure, and excessive inflammation. Another aspect which is emerging, and I'm sorry for the very complex picture, but it just, the message of this one is that one of the characteristics of this virus and all coronavirus, all the highly pathogenic coronaviruses, in fact, is that they will block very efficiently a very early innate immune response, which is the production of interferon. And interferons, they will basically block virus replication and protect the cells from the virus itself. These viruses have, through different functions, and we in our lab, we are particularly interested in the function of the proteases. They will both completely block interferon production and also activate a pathway for production of a different kind of inflammatory cytokines, which are actually responsible for the very early, or for the late phase of the disease, which is characterized by this excessive production of cytokine, excessive inflammation, and all the deadly consequences that come with that. I think I will stop here, and eventually we can go back and look at some of the details if there are questions on that.
Thank you, really. Thank you so much. And thank you for your very nice presentation, Maria Grazia. I personally enjoyed really very much. I'm sure that we gave so many new ideas that we can work on for many years. Thanks. But I would like to ask just if someone want to know something, one or two questions, not more than that, please. Can I read? Can COVID-19 virus potentially turn into a form of cancer?
Should I answer this one, Alberto? I would say that based on current knowledge, no, there is not directly, at least, because what makes a big difference between the viruses that cause cancer and the viruses that do not cause cancer is the capacity of the viruses that cause cancer to promote chronic infections. And this long-term infection is what requires sort of to accumulate genetic changes that will end up becoming a cancer. And fortunately or unfortunately for us, unfortunately in terms of acute response, and fortunately in terms of cancer, this appears to be a very acute infection. And it is very unlikely that it will cause cancer directly. Although, as we are learning, there are many possibilities. And the chronic inflammation, which could happen, could be indirectly be associated with cancer. But fortunately, it will take at least many, many years before we see this potential complication, I guess.
Thank you, Maria Grazia. Do we have any other question? What is the current knowledge of so-called resistors, people that seem born with a natural immunity?
This is yours, Alberto.
Yeah.
Yeah. Well, it's an easy answer. We don't know. In other words, we know that there is a genetic risk related to various locations on the chromosome, in particular chromosome 3. There is a story about genes that we inherited from interbreeding of Homo sapiens with Neanderthal, and which may have endowed us with genes that increase some kind of inflammation, but on the other hand, it may have been an evolutionary advantage. Otherwise, we would not have kept these genes, we assume. I think, Maria Grazia, you would agree with that, I guess.
Absolutely. I must say, I think that Neanderthal is still a mystery for me.
Yes. So we really don't know. For instance, let me take the most extreme situation. We have two people. I mean, females are relatively more resistant, but what is really striking are children. It's completely different from, say, influenza. And why is that? Personally, I don't understand. There may be an issue of the receptors that Professor Mazzucci beautifully described, the expression levels of the receptor. Of course, the immune system of children is better than my immune system. That's pretty obvious. The third explanation or third component is that they have children always constantly go to a fitness room, the fitness room of kindergarten, the fitness room of vaccination, constant vaccination, and trained innate immunity. We really don't understand, but this has been really something very good in this pandemic, that the very fact that children, but in essence, we don't know.
I have actually another example, which is a little bit more puzzling, because we know that children sort of they can cope with viral infections in general better than adults. I mean, all the children diseases in children are okay. If they occur in adults, they are very fun. They are very dangerous. But my sister lives in Bergamo, and they have lived through the pandemic, and none of the family actually was sick, which was very good. But anyway, they all tested for antibodies, and it turns out that my sister's husband is positive and no one else in the family. So how did they manage? There is so much that we don't know.
Yeah, that's totally true. I want really to thank once more all of you, Riccardo Villari, as president of the foundation IDIS-Città della Scienza, and Silvia Bencivelli for the beautiful movie that we have seen, and of course Alberto Mantovani and Maria Grazia Masucci for the beautiful presentation that also people that don't know too much could understand, at least have a feeling of what you are saying. And we enjoyed really so much your words. But last but not least, I want to say thank you very much to Mattia Mazza, the Italian embassy in Sweden, that have organized this meeting today. And I'm really feel that we should strengthen the collaboration between Italy and Sweden. There are many points of contact, and I think that is a good occasion to try to start also with perhaps a bilateral agreement in research between our countries. Thank you so much, and have a beautiful day, evening, let's say. And thank you for being with us for so long.
Arrivederci.
Thank you. Arrivederci. Thank
Ciao,
you.
buonasera a tutti.
Ciao.